First thing I'm going to do is forget to turn my microphone on, so I'm going to not do that. Can you guys hear me okay? Yeah. Yeah, okay. Hi, everybody. Hi. Thanks for coming tonight. It's a real honor and a rare honor for scientists to get to speak to people that aren't other scientists. There might be other scientists in the room, but generally when we get up and talk, you know, we're at a conference with a room of people that we've known for years because we do similar things. So as exciting as that is for me to get to talk to you, I want you to call me out if I start going into my old, jargon that I'm going to try not to do. But if you need me to explain something that I've glossed over, it's going to be informal tonight, so just stop me and I'll clarify anything. So what I'm going to talk to you about today, I'm going to start out by talking a little bit about how humans have changed the planet and how some of that has impacted vector-borne diseases. And then we're going to talk a little bit about just mosquito biology and disease transmission in general. And then I'm going to tell you about a few of the research projects that I've been working on the last few years, looking at asking questions about how climate change, species invasions, and urbanization are impacting mosquito communities. So we're living right now in what people have dubbed the Anthropocene, which is, so you've heard of the Pleistocene, right? And so the idea is that humans have modified the environment, the whole planet, to such a degree that we're in our own epoch. But I've got examples up here of massive urbanization, industrial agriculture, deforestation, and something that I've been thinking about a lot more recently is the fact that we've taken away night. Yeah, light pollution, it's huge, right? And so all of these ways that we have, greatly modified our planet has led to a lot of consequences. So, I'm not going to make us all super depressed today. I mean, I think we all know about a lot of stuff that's going on, but, species extinctions, biodiversity losses, the acidification of the ocean, these all have consequences for ecosystems. But one of the important consequences of all of this re-engineering of the planet that we've done that has direct impacts on us as humans are emerging infectious disease events. Okay, so what an emerging infectious disease is one of three things. So it's either an exotic pathogen that's introduced into a new area it's never been before, based on our trade and our travel. So we're moving things around on the planet all the time. So we've introduced new diseases. So West Nile virus is an example. That was never seen in the United States until 1999. Zika is another example. That was a virus that we discovered in mosquitoes in 1947. So we knew about it, but it wasn't until the 2000s and especially in the mid 2010s that it spread all over the globe. All right. Another way that emerging infectious diseases can happen is when native pathogens that we knew are present in an area, suddenly we have major outbreaks of them and major increases in the number of cases that we've seen. An example of that was the reforestation of New England. So we let the forest grow back up. That caused the deer population to increase, which caused the tick population to increase. And though we were then in contact with these ticks that were infected with this bacteria that causes Lyme disease, that had always been there. Just when we changed the landscape again, it increased in incidence. And then there's a third category of emerging infectious diseases, and that's when we just find something new that we've never seen before. And this happens a lot with, so some recent examples are bourbon virus, which is spread by ticks. But we just didn't know it existed. And a lot of times we come into contact with these when we start encroaching into habitat that we don't have any business being in, right? Yeah?
That's the second picture.
This one?
Yeah.
That's a baby with microcephaly from a Zika-infected mother. So all of the examples that I've given you today are vector-borne. I'm going to define vector-borne as it's a pathogen that's transmitted from one organism to another via a third organism. And generally that's going to be a biting arthropod, so a tick or a mosquito. So that's what a vector is. And this paper came out in 2008, and it's showing that the number of emerging infectious disease events that have happened over the last 60 years. And we can see, as we move further into this human epoch, this Anthropocene, and get closer to the modern age, we're getting an increase in the number of emerging infectious disease events. And this white bar here on the top, those are the percentage that are due to vector-borne diseases. So they're a big portion of the new diseases that we're seeing. And this paper ends in 2004. And since 2004, we found 9 new pathogens that are transmitted by ticks and mosquitoes. So this is an ongoing and current problem. Now, not only are we finding new vector-borne diseases and moving them around the globe, we're just in general seeing more cases of vector-borne disease. So the CDC put out this report just last year, and this is showing you just the number of diseases, number of cases of vector-borne disease in the US. And since 2004 to 2016, we have tripled the number of cases that we're seeing. So there's really something going on here. Now, I'm going to talk about the US mostly, just because this is where I do my work, and this is, the area of the world I'm the most knowledgeable in. But vector-borne disease burden is not distributed equally globally. So on this figure, the darker the color the country is, the higher the burden of the global vector-borne diseases that we see. And so even though in the US, that's horrifying, this tripling of cases, we still are the lowest incidence of deaths by vector-borne disease in the entire world.
Mostly malaria?
A lot of it is malaria, yeah. But there are a lot of viruses that can also be fatal that are transmitted. So just globally, to give you an idea of the scope of the problem of vector-borne disease, and this includes vector-borne diseases from all arthropods, not just mosquitoes and ticks. There's approximately 1 billion cases, 1 million deaths, and 17% of every infectious disease that's transmitted on this planet is transmitted by a vector. Now, the reason that there's so much vector-borne disease is because these insects and other arthropods, they literally get into our bloodstream, right? We call it the blood-feeding habit. So by directly injecting themselves into our blood where pathogens are flowing freely, it's not surprising that we've seen so many vector-borne diseases. And in fact, blood feeding has evolved 6 separate times in the history of arthropods. So this is a phylogenetic tree of all the arthropods. So over here, and everything that I've circled in red here is a blood-sucking arthropod. So we've got our bed bug is over here, which is more closely related to the flea. And then we get our mosquitoes over here and ticks. So the blood-feeding habit is something that's evolved 6 different times. So it's a pretty effective strategy.
Go back. Okay. Is that a moth?
Yes. There are a couple of species of moths that do suck blood, yes. Mm-hmm. Okay. So just to give you a little bit of background on the sort of the theory behind disease transmission in general, this is called the epidemiological triad. And so it's a really simple visual model to get at the point that in order for a disease event to happen, you need to have a pathogen, a host, and an environment that's suitable enough for both of them to come together in the same place at the same time. Now with vector-borne disease, you're also going to have the environment's also going to have to support a vector. Now, this is a lot more complicated than an infectious disease that's spread, like the flu, person to person. All you need is to have 2 people in one space at the same time where one is infected and one is susceptible. Now, we need to have an environment that's suitable for this mosquito, the virus, and in most cases, vector-borne diseases actually don't have humans as important reservoirs. They don't replicate in the human enough to contribute to the transmission cycle. These are called zoonoses. That's this word I have here. And what that means is that which is West Nile as an example. So what that means is that West Nile actually is a pathogen of birds and mosquitoes. So it circulates here in this cycle. birds are able to replicate the pathogen at a high enough load that a mosquito can, imbibe the blood and the virus at a high enough dose to become infective again. And people and horses, we're dead-end hosts. So we're just kind of incidentally there, accidentally infected. Mosquitoes transmit viral pathogens, protozoan pathogens, so that's malaria, and also nematodes, these little worms. So they can transmit all kinds of different things. And what's amazing about these pathogens is that they have to be able to replicate and grow in both human tissue and animal tissue often, and also mosquitoes. So think about, you get a cold, your cat can't get it, right? So there's often such specificity in what a pathogen can replicate in that we don't see so much, so it's really incredible that all these different classifications of parasites can replicate in both insects and humans. I'm going to bring back this vectorial capacity model and just talk a little bit about some of the things that are important when we determine your risk of getting an infection. So this is a theoretical model, but you can parameterize it. So in order for all these things to come into contact so you get that disease case is what you're interested in, you have to have a population of mosquitoes that's pretty dense, so you're getting multiple bites. You have to have a mosquito that's feeding frequently enough that it lives long enough to take multiple bites, because when it first takes that infectious blood meal, let's say it finds an infectious blood meal, the time that it takes that blood meal till the time that it's infectious again can be anywhere from 2 days to 2 weeks. Because they take, so let's take a virus, for example. So I ingest a virus in my blood meal. I have to, if I'm that virus, I have to escape out of the gut. And then I have to replicate in all of the tissues of the body of that mosquito and make my way to the salivary glands so that I can then be injected into my next victim. And that takes time. So how long a vector lives is going to be important. If you only get that one meal and you die 7 days later, you may not ever be infectious. And another important thing to think about is vector competence. And what that means is that a mosquito is even actually able to become infected. So not many species transmit diseases. They have physiological barriers that won't allow the virus to escape out of it. So all these things are really important in determining why we see vector-borne disease where we do, because we don't see it everywhere, right? So underlying all of this stuff, there's the breeding, all these things need to be available that are necessary to have all these things come together at once. And even though, like I said, we're not necessarily important hosts, we're dead-end hosts for pathogens, there's lots of factors about us that will determine whether we see disease or not. For instance, what's our socioeconomic status is found to be incredibly important in your risk for vector-borne disease. Your age group, so for West Nile virus, people over 60 are much more likely to see disease. For La Crosse virus, people under 10 are much more likely to get disease. So there's human factors that matter too. And your behavior. Do you hang out outside all night without bug spray on? Or do you stay inside with your windows and your air conditioning, right? Yeah?
Why would children and older people, because they...
It has to do with their immunological status and properties of where the virus replicates. So, okay. So, there's also a lot of environmental factors that we know impact replication of pathogens in mosquitoes. So for instance, we've known for a long time that temperature is going to influence a mosquito's ability to transmit pathogens. So we're looking at the percent disseminated over here. That's science speak for it's able to transmit it. And over time, and these are at 4 different temperatures. So at 30 degrees Celsius, which is, like 88 degrees Fahrenheit, just in a matter of days, that mosquito is able to transmit it. But at, say here, 20 degrees Celsius, very few of those mosquitoes are going to be able to transmit it. There's some more interesting ecological stuff going on with mosquitoes and their ability to transmit pathogens. So here we're looking at larval competition. So mosquitoes are aquatic as larvae, and they're stuck. They can't get out of their habitat. And if there's not very much food in there and lots of mosquitoes, they compete aggressively for those resources. And that larval competition in the immature stage can affect how much a mosquito can transmit a pathogen as an adult. So we see that, again, the higher up here and the higher here is the more likely they are to transmit. And these are different densities of larvae. And the two highest densities here, if they were reared under high densities, they're more likely to transmit a pathogen, right? Now, this is kind of a newer thing, and this has been really blowing my mind lately, is that we've been discovering that the microbiome of the mosquitoes can influence their ability to transmit pathogens. You guys have heard of the microbiome. It's all the guts, the bacteria and stuff that live in our guts, and we're learning more and more about how it's essential for human health. It's also essential for mosquitoes. So there's a lot of interactions between things that mosquitoes transmit and just the natural bacteria in their guts. And this study found that people who are currently taking a dose of antibiotics, when a mosquito feeds on them, they also inject that antibiotics in the blood meal. It kills their microbiome, and it changes their ability to transmit pathogens.
Does it increase it or decrease it?
It depends on the pathogen, but it generally increases it.
Because it gives it room not to compete with the...
Exactly. So it's not competing with the microbes for space. There's a lot more complicated stuff going on with it, with proteins that are produced and stuff, but in general, it increases their ability to transmit. Tell that to your doctor next time he wants to give you antibiotics when you don't need them. Okay. So let's talk about mosquitoes just in general a little bit. So there are lots and lots of different kinds of mosquitoes. And I'm an ecologist, okay? So I work with a lot of people that study patterns of diversity, you know, with trees or plants. And, you know, I tell them about I work with mosquitoes, and even ecologists are like, oh, isn't there just like one mosquito? You know, they don't even think about the diversity of them. So there are worldwide 3,500 species. They have all kinds of different life histories. Some breed in ponds, some breed in pitcher plants. They have different feeding strategies. Some feed during the night, some feed during the day. Some will only feed on one or two specific host species. Some will feed on anything. And they all vary, again, in that important vector competence that are their ability to transmit pathogens. And so because of this, there's actually very few species that are good vectors. Here are three of the globally most important. This is Aedes aegypti. This is the yellow fever mosquito. This is a culex that is responsible for a lot of the West Nile and filariasis. And then Anopheles gambiae, who's responsible for a lot of the deadly malaria transmission. And the things that are common about these species that are good vectors is that they're physiologically, they can transmit it. They don't have a block that won't allow them to. They're associated with humans, okay? So they're not forest species that we rarely encounter. And in some cases, we could even call them human adapted. So they're literally adapted to living with us and no longer live in like natural habitat. And they also have the right host preferences. So they're feeding on species that can transmit things back and forth. So they're not, you know, taking only blood meals from lizards, right? So just to make sure that we're all on the same page about what the basic mosquito life cycle is, they start out as eggs. This is an egg here. You can see how small it is. And these, just depending on the species, some are laid on the water, some are laid along the sides of containers. But they're all laid in water. They then have our larvae, and a lot of the work that I do looks at what happens in the larval habitat. So they have 4 instars. So they molt 4 times in the aquatic habitat. In the aquatic habitat, like I said earlier, they're stuck there. They can't get out. They can't choose why they're there. And their food source is the bacteria and the fungus and the other protozoans that live in the water and break down detritus. So there's very little primary productivity in these aquatic habitats, so the food base comes when things fall in and decompose. So leaves fall in, a bug goes in and drowns. And so remember that detritus and the kinds of detritus in a habitat is important, because a lot of my research will be about that we'll get to in a minute. So if they make it through these larval feeding stage, they then pupate, where they no longer feed, but they're still aquatic. So this is like the equivalent of a butterfly's chrysalis, okay? So they're metamorphosizing into the adult here. And then if you can see this picture of them emerging out of the water and becoming a terrestrial adult. So I think that blood feeding because they're blood feeders, that's why people care about mosquitoes, right? So just to tell you a little bit about, just briefly about their blood feeding, this is their proboscis is what it's called, their mouth part. It's like a needle. Here's a scanning electron micrograph picture of what that looks like. So if you can tell here, she has a sheath over her needle-like proboscis that she lifts up and injects right into your capillaries. And the reason that mosquito bites itch is because she's injecting her saliva into you, and her saliva has proteins in it that stop your blood from clotting, that are pain relieving, so you don't notice her as fast. Anesthetics, so that's pretty interesting. So then your immune system comes and attacks these proteins, and you swell up and you get an itch. Mosquitoes also, this is a picture of a fully engorged gut that I dissected out of a mosquito. And here you can see just how much her abdomen will expand. She can ingest up to 2 to 3 times her weight in blood in one meal. And I don't know if you've noticed, but I've started saying she now, because only female mosquitoes take blood meals. They use the protein in the blood to make their eggs. So male mosquitoes don't take blood. And in fact, both male and mosquitoes are nectar feeders. So the first 5 days or so of her life, she won't even be interested in a blood meal. She'll just be running around getting nectar. So they're actually pollinators, if you wanted to look for a reason to give them a little nod, give them a little credit, you know. I joke, but actually these are some of the most fascinating ecological creatures that some of my colleagues don't agree with me, but there's a lot of us that make our living studying their ecology. Okay, so what I study a group of mosquitoes that are called container breeding mosquitoes. It's not creatively named. They're called that because they breed in containers, okay? Ancestrally, they bred in like tree rot holes that hold water, okay? And I still do some work with these, but they love our trash. So these are pictures of, experimental units from things that I've done. So you can just put a cup out in the woods or in the city and they'll come to you, pretty easy system to work with. They also breed in stuff that we leave out. So abandoned tires are a huge thing that they breed in. This is a cemetery vase. So the cemeteries have caught on to this. They don't really let you leave water holding containers anymore. But anyway, so these container breeding mosquitoes are an ecological group. There's I work with probably 4 species, and I'll introduce you guys to them in a little bit. But these containers are discrete ecological communities. So everything that you think about, like ecological interactions between species, they happen in these containers. So there are competitors, like I mentioned, there's resource competition going on in there. There are predators in here. So mosquitoes have enough evolutionary history with predators that they change their behavior in response to predators to lessen their likelihood of getting eaten. And there's also parasites in these. So not things that mosquitoes transmit necessarily, but things that are parasites of mosquitoes. And these are really interesting to work with for a couple of reasons. One, they're really easy to manipulate. So you can go out and you can put 50 buckets out at one site and the size of this table, and you have 50 replicates for your experiment, right? So that's really great. And everything that we learn about them, whether we're trying to answer basic ecological questions about predator-prey interactions, it all has an applied aspect to it too, because they're disease vectors, right? It's a great system. So I don't actually study right now the diseases that they transmit, okay? So I don't forecast, disease transmission among humans. I'm interested in mosquitoes because of vector-borne disease. That's the base of my interest. But when I started studying like public health and vector-borne disease epidemiology, I realized this is inherently an ecological problem, okay? Because of all the different players that need to come in one time. So that's why I started studying ecology. Yeah?
How long does a mosquito live?
That's a great question. From egg to adult, probably 2 to 3 weeks. But again, that's really going to depend on the ambient temperature, ambient humidity, how much food it has access to. I've had mosquitoes live in the lab under great conditions for 4 months. Yeah, that's annoying because you got to check them every day. Believe me, I wish they would have died. But in the field, probably a few weeks under field conditions. So the work that I'm going to tell you about now, and why I framed my introduction the way I did, is because I believe, so classic ecology is in the woods, it's butterflies, it's pristine prairies, we know, but that's not the world we live in anymore, right? The world we live in has completely been re-engineered by humans. So as an ecologist and as a disease vector ecologist, I frame the questions in my research in terms of how different human impacts on species are impacting mosquitoes, okay? And so some of the most important things that we have done to this planet is we're changing the climate, and so our work looks at how warming temperatures are going to affect mosquitoes, as well as how they might be adapting to climate in a new range, as well as species invasions. A lot of the mosquitoes in this country are actually invasive, and how urbanization is also impacting mosquitoes. So I don't do any of this work alone, so I need to acknowledge the people that I work with before I start getting into our projects. So first I'd like to acknowledge Tyson Research Center, which is not far from here. It's just down 44, one exit. And Dr. Kim Medley, who is my boss and collaborator, and all the work that I'm going to tell you about today, I do work with her. And then 2 of my collaborators that I work not so closely with, but are still very important in forming my work. And Dr. Soli Adelson has given a talk here before. Actually, I don't know if any of you were here. She studies ticks. Anyway, so those are the people that I work with. These are the species that I work with, even though my friends make fun of me because I refer to mosquitoes as people. That's just how much time I spend with them. So the stars of the rest of our show today are the Asian tiger mosquito, Aedes albopictus. This is one species that I'm afraid as I talk, I'm going to go back and forth between referring to it as the Asian tiger mosquito and albopictus. It's the same thing. It's just a common name and a species name. You guys are probably all familiar with this species. This is the black and white stripy one that bites you during the day that wasn't here 25 years ago. And then I work with a native mosquito, the native eastern treehole mosquito. She is a forest-dwelling species. We rarely see her in the city. And then a second invasive mosquito that's a newer arrival, the Asian rockpool mosquito. And then I'm going to tell you today about a parasite that we do a lot of work with. They're called Gregorine parasites. These are not things that mosquitoes transmit. These are protozoan parasites that live in the guts of mosquito as larvae. And they're really easy to manipulate. So I can do experimental infections in the lab. So I use them to ask just kind of basic questions with disease ecology, okay? So we're going to start with the Asian tiger mosquito, Aedes albopictus. She's a beautiful mosquito, shiny, stripy. And she is actually considered one of the 100 most invasive species on the planet. So again, I'm going to focus on its invasion in the United States, but it is spread globally, and in the last like 35 years. Okay, So this is its current range in the US. And this mosquito she's, nothing can mess with her, basically. She'll feed on anything. She lets me keep them in colony in the lab. She's an incredible mosquito. Now, she got here, right down here, where this star is.
Goes wrong in Houston.
Everything goes wrong in Houston. They're famous, right? So Harris County Mosquito Control in 1985 discovered a breeding population of this mosquito. These blue, those are counties that the mosquito was discovered 1 year later. Okay. Yeah, they did. They came straight to St. Louis. We are like not ground zero, we're ground one for this invasion. So they just spread so rapidly. In 2001, this is what the distribution looked like. And don't be fooled by the Arkansas. Arkansas is embarrassed. They never did any collecting. They were in Arkansas, okay? So in 2001, it kind of looked like it had filled in its range. It hadn't moved much in the last 10 years, right? But then, in the last 15 years, this is what the distribution looks like now. So we had kind of a quiet period where it wasn't moving anymore. And now it's filling in areas up here in the north, and it's rapidly spreading just in the last 5 years in Southern California. They don't know what to do with themselves. They're having such a crazy infestation now with this species. And there's evidence that this range expansion to the north it's definitely happening, and these are established populations now. And so the reason that the species was able to move so quickly across the US and the globe, really, is because we are moving it, all right? So this is just, our trade and travel, just from a couple years ago. So we are moving things all over the place. And so some of this movement of species is, okay, an individual mosquito gets in your car and you drive a neighborhood over, and when you open the door, it flies out, right? That happens. And these mosquitoes do disperse naturally, but not very much. So the maximum distance that an adult one of these mosquitoes will fly in its lifetime is 0.6 miles. Okay, so they're not moving on their own. How we're moving them is in our trash. Okay, so this is not a picture I took, but I spend a lot of work days in areas like this. So it's, I'm not a glamorous ecologist that works with dolphins or anything. I'm in the trash heaps, all right? And this is just classic. You see these abandoned tires laying around with other dump stuff, and it's shaded here. They like it a little shaded, and it rains, and these tires just hold that water. And these are just nice containers for the mosquitoes to, lay their eggs in. And they lay eggs that are desiccation resistant. So if it dries out, the eggs are still in there when it refloods, and maybe we've moved them, somebody's picked them up and moved them to another place, a dump site, right? There's a huge used tire trade. People grind up the tires and use them different things. So we're always moving these mosquitoes all over the place because they breed in our trash. Okay. So we were interested in looking at, we're seeing this northern range expansion. So we wanted to know, okay, are they expanding further north than we would expect based on their distribution in their native range? So this was work done by my collaborator and boss, Kim Medley. I wasn't involved in it, but she did what's called a reciprocal distribution model. Okay, so this is Albopictus, the Asian tiger mosquito's native range. All these dots are occurrences, and the darker it is, the more likely it is that you're going to find them there. Okay, and this is based on 18 different climatic variables. And so what she did is she put that modeled that on the U.S. Okay, based on the climate factors. So if this mosquito its range is in the exact same niche as where it originally came from, it would be distributed like this, okay? And if you remember what this distribution map looked like that I showed you, this model failed to detect about 20% of its actual range here. And so the question was, well, okay, has this mosquito evolved since it got here to be able to expand further in its range than it would have. Yeah?
Or is it just getting hotter?
That's my next slide. Yeah. What was your question?
Or is it just getting hotter?
And that could be it too, right? It may also be that it's getting warmer. So, especially with the northern range, which is what I'm going to focus on for this portion of my talk, the most important climatic factor is it able to withstand winter? Okay, and how this species survives winter is in the egg stage. So this is a big blown up image of an Asian tiger mosquito egg. And this is a blown up image of an Asian tiger mosquito egg that's been bleached so you can see the embryo inside it. Okay, so they survive the winter by going into what we call photoperiodically induced diapause. And so what that means is it's a state of hibernation that they enter into in response to declining day lengths. So day length is a reliable indicator of impending cold, right? So mosquitoes are not at all the only species that respond to day lengths. It's very, very common. So a female mosquito will sense that the day lengths are shortening. And instead of producing embryos that directly develop, she will produce embryos that are not developing. They have much reduced metabolism. They have a higher content of lipid in them so that they have more reserves. And they're also coated in this like waxy substance that helps them from drying out. Okay. In St. Louis, this is predicted to start happening based on our latitude in like a few weeks, even though it's going to be hot for a while. Okay. So some other stuff that we know about this diapause trait is that it's under really strong selection pressure. So when the mosquitoes arrived here in 85, all of them could diapause. So we figured out that they were from a temperate population in Japan. So they all could diapause. And so we followed them, not me, but other research followed them and found that they were quickly losing this trait. So they were rapidly losing the ability to diapause as they spread south, where it doesn't get very cold. So this is latitude on this x-axis here. 24 is Key West, 42 is Detroit, and on the y-axis here is diapause incidence. And so that's the percentage of the population that will go into diapause. This line here, this was data that was collected on populations from across this latitudinal gradient in 2008. So we see that at these lower latitudes, we've got less diapause, okay? So maybe 80 to 60% of mosquitoes are going into diapause. But then 8 years later, only 8 years, we see this climb latitudinal in diapause. So they've just so rapidly lost the ability to diapause, okay? So we know that this trait must be under really strong selection pressure for them to evolve that quickly, okay? So what we wanted to know was this Asian tiger locally adapted to US winter? So we have evidence to suggest that this diapause trait, which helps them withstand winter, they can rapidly evolve, right? So the idea is, are these populations up here at the range edge where it's getting almost too cold for them, but they're still there, are they going to be able to survive winter better than these populations from down here in the core of the range where it's warmer? So I also want to point out, other researchers had saw this diapause. We know diapause helps you survive winter. We know that diapause is selected upon, but nobody's actually tested if these populations actually survive better, okay? So we were going to test out, have these populations adapted? So how we did this is we sampled a bunch of populations here and at the range edge and the range core, and then we established colonies in the lab. And I'll just briefly show you what establishing a mosquito colony means, because I spend a lot of my life doing this. So you get these eggs and you hatch them out. These are a whole bunch of larvae, cute little wigglers that you see running around in water. And I just rear them in mass pans in these environmental chambers where I can adjust the day length, the temperature. And then they get, the pupae get transferred into these like vials these flasks. You can see all the adults in there, and then I aspirate them into these cages. And then I can use, I can order blood that's been defibrinated so it doesn't clot, and put it in like a casing and blood feed them that way. So I can just create generation after generation of mosquitoes in the lab. And then I collect their eggs on these little papers, and I can store them. And as I did in this experiment, I can mail them to Wisconsin and see how well they overwinter, okay? So I established these colonies in my chambers where I can shorten the day length. I made the day length real short. I induced a diapause. I collected those eggs, and then I sent them out into the field to endure an actual winter. So one group of the eggs went to, I believe it was Huntsville, down here, in the range core, where it's nice and pretty warm. One group went to the range edge, out here in Harrisburg, Pennsylvania. One group went beyond its current range, just to see how well they would do. And then the final group stayed in my environmental chamber right here as a control to make sure that a whole bunch of them just don't die off over this 3-4 month span anyway, right? So they're kept under ideal conditions. And the idea is that if range edge populations do better in the range edge than the range core populations, that they're adapted to it, okay? Does that make sense? All right, excellent. Okay, so we recorded the temperature the whole time while we were doing it. This is Wisconsin, Pennsylvania, and Macon. So we were able to show that yes, there are definitely differences in the average temperature. It's definitely colder up there. And what we were able to show was that these mosquitoes are in fact locally adapted. They have adapted to winter. So this is percent survival of the mosquito eggs that we sent out. These, the black circles are the populations from the core edge, from these guys here, and the blue are the edge populations. When they were kept in the chamber, my nice control, basically everybody made it the 3 months that they were there, okay? When they were in the range core, they did the same. So everybody, you know, did pretty good down here in these nice winters. But when they went to the range edge, we had significantly higher survival in the edge populations than the core populations, which is exactly what we would expect if they are adapting to winter in their invaded range. So this has implications for their further range expansion. If just, you know, in the 30 years that they've been here, they've adapted to our conditions, you know. I will point out that not a single mosquito survived in Wisconsin. Wisconsin's happy to hear that. I know some people that work on mosquitoes in Wisconsin, and they are creeping up in here now. But at least in 2015 when we did this, nobody survived this far up. So they have a while anyway.
It doesn't look like they had too great a time in Chicago either. There's that little notch right there.
Yeah, they do maintain a population in Chicago.
They're up there,
yeah, they do. Mm-hmm.
They didn't like the winters either.
No. Chicago's a whole nother story. But anyway, so to conclude this section, these mosquitoes, these invasive mosquitoes, are spread across the country and the globe by us. And they're evolving as they go. Okay, so what we knew about them in their native range isn't going to hold in their invasive range. And because we know they're evolving this rapidly, there is potential for future climate adaptation. Okay, so as we warm, we're going to see probably movement north anyway, just because it's warmer, but we're also probably going to see them adapting to warmer temperatures as well. So, and I think this is an important point because a lot of times when we talk about vector-borne diseases, we're talking about vector-borne diseases tonight, but we neglect to take into account that they could evolve, okay? So we just say, oh, they can't be here because they can't make it here now. You know, so the fact that they can evolve rapidly hasn't really been part of the discussion. So there's important implications. So I talked about the scale of Albopictus on, you know, the scale of the country, okay? But on local scales, their distribution is also different. So, you know, just at the scale of a city or a neighborhood, their distribution is going to be dictated by things like the microclimate. So it's cooler under a bush than, you know, out in the direct sun. the availability of blood and sugar sources for the adults to feed on, the availability of larval habitats, and community level interactions like competition and predation. So one of the things I haven't told you yet about Aedes albopictus is as it spread rapidly across the country, there were local extinctions of resident species. So this, I told you she's just an incredible mosquito. She's such a good resource competitor that local populations just were gone. Within 2 years, they went extinct.
Is that the larval competition?
Exactly. Yeah, they compete in the larval habitat. They don't really compete as adults. And so theory said when they looked at these natural habitats where some of the resident species were more common than these like artificial tires, they did some experiments and they showed that in artificial habitats, albopictus, the Asian tiger, should probably outcompete and get rid of the natives, but not in these tree hole containers. That there's something fundamentally different about the two kinds of habitats that they would not get rid of them here. And also part of that might have been that the Asian tiger is pretty vulnerable to predation compared to native species. So it gets eaten a lot more likely than native species. So that being said, I've been working out at Tyson Research Center since 2012 and using these artificial habitats that albopictus should kick out the native species in, all right? And I've had established habitats since 2013 of all different sizes. So they should be doing fine. And they should be the only mosquito left, right? But what's actually happened in my years of working there is that I feel like there's fewer of them than there used to be when they should just be taking over. So they do show up every year, but they show up like in August, and they never get very dense. Okay, so that's made me wonder, well, what's happening along this land use gradient that's making it that so that we're not seeing lots of albopictus in artificial containers in the woods. But I had to kind of start at the beginning because, believe it or not, nobody's been studying mosquitoes in St. Louis for like a long time. So, and there actually is a mosquito-transmitted pathogen called St. Louis encephalitis virus, but nobody's been studying anything here for a while. I don't know, you guys were famous for like a minute.
It was a big deal for a while.
A long time ago.
They stopped?
It's rare. It's rare.
Didn't have the gravitas to be a thing anymore.
I'm not going to lie to you, I don't know a ton about the reason that we don't see very much of it.
Is it possible that the trash in the urban areas that keep the dragonflies away doesn't exist in the woods and there's more predation in a more pristine area?
That would be a hypothesis, yes. I don't see very much predation in my artificial containers in the woods. So that would be, an immediate thing to think, but it's just really not that high. It is very high still in the tree holes in those natural habitats, but we just don't see that much of it.
That's what's getting them there,
Yeah, exactly, yeah.
Something's wise to them in there.
Yes, yeah. I have a data set looking at that I won't have time to talk about today, but. Okay. So there's lots of things that are different along this urban to rural land use gradient, right? So here's a picture of I knew I was going to do that. An urban backyard, and then you've got this nice suburban backyard with all these flowers and shade and stuff. And then here at the woods, it's very different, right? So there's lots of things that are different along this land use gradient that could describe, why part of the reason why we don't see a lot of albopictus out in this area. So we needed to set out. Number one, we needed to know that in fact, because I assumed, but I didn't know. I needed to set out and see that there's seasonal and abundance patterns across the city of St. Louis and into the woods were different, okay? So are there more mosquitoes? Do they come out earlier in urban, suburban areas than they do in the woods? I needed to confirm that. And then I wanted to know to what extent temperatures differ. There's an important urban heat island. Have you guys heard of the urban heat island? So I wanted to, and because mosquitoes are, cold-blooded ectothermic organisms, temperature is very important for them. So I wanted to see, in the St. Louis region, how much do the temperatures differ between the land use types? And then I was really curious about whether the artificial larval habitats differ in quality, okay? So remember I said detritus is important, and the type of detritus that falls is also important. So I thought, well, maybe these slow decaying oak leaves that fall in the habitats in the woods can't support lots of the Asian tiger mosquitoes versus like the nice juicy rhododendron flowers that fall in somebody's suburban yard. You know what I mean? So I wanted to see if perhaps just the quality of the larval habitat was bad in the woods. All right. So we set out and we did this for 2 years. We had 4 sites in the city, 4 in the suburbs, 4 to 6 in rural locations, and we set out my classic black cups, and we recorded temperatures. And every week for 2 years during the summer, we, mainly my students, pictured here, went out and collected all the larvae in them. We had to take all of them because the nice friends and family that allowed us to use their yard didn't really want mosquitoes flying around, so we had to take them every week. Refilled the water levels, and then we identified who was there, all right? And so what I'm showing you here, this is just 2018 data. We started to see mosquitoes in May, and this is the number of Albopictus with urban areas, suburban areas, and rural areas. So my suspicion was correct, okay? They're here, they're in the woods, but they don't show up hardly at all for, what, a month after they show up in the city, okay? And they just have like really reduced abundance. And again, this is in artificial containers there were no predators in this study. And just in general over the 2 years, we didn't find that the number of Albopictus was really different between urban and suburban. They do great, doesn't matter urban or suburban, but they just consistently both years were the same. They just don't do good in rural areas. Now, just to show you the temperature data, this is an ugly figure, but I just want to demonstrate to you that the red is urban and the rural is green. Urban and suburban are different, but often not statistically different. So that we see some days where we got 28, 29 here and 33 here. So some days there's 3 to 4 degrees difference between the woods and the city. And 3 to 4 degrees difference for a developing mosquito is huge. So that'll make them go from an egg to an adult in the span of like 6 days versus like 11 or 12 days. Okay, so that's really going to influence population growth. So I think, I don't have the data to back this up, but I think part of the reason that they're doing better is because it's warmer and they develop faster, so populations grow faster. Now, so get back to the question, does the artificial habitat differ along these gradients? In 2018, I bagged up all my cups, all my cups here in bags, and I brought them back into the lab. And I did a bunch of water chemistry. So things like tannins, nitrogen, pH level are probably important for mosquito development. And then I added an experimental cohort of first and star Albopictus larvae to each habitat to see do they actually have better survival in habitats that were created along this land use gradient. And just to give you an idea of what 3 months in the field collecting detritus looks like, so this is the kind of stuff you can see how tanned the water has gotten and all the different decomposing. This is Japanese maple. And here in the rural areas, you got these big old like oak and whatever, that elm. I'm not good with my plants, but there you go, sure. I'm not good with my plants. Anyway, and so what we found was that with tannins, there wasn't any difference this is the increasing tannin level. They weren't different, but nitrogen was different. So there's higher nitrogen levels in artificial habitats in rural areas. pH is higher in urban areas. And that we also saw that the volume of water in the habitats was different. So in urban areas, there's a faster water desiccation than there is in rural areas, which is I'm not surprised by, but it's good to have the data, and it's likely due to temperature. So there's desiccating faster at higher temperatures. But this is the proportion of that cohort of Albopictus that I added. There was absolutely no difference in how they survived. And how well they survived, again, percent larval survival, wasn't related to the number of mosquitoes that were collected in an individual container. So if there was a lot of mosquitoes that we collected over the year in one container, that didn't mean that they survived better. So to conclude this part, we do see delayed phenology and reduced abundances of the Asian tiger in rural habitat, maybe partly due to warmer conditions in urban and suburban areas, but it's not due to any part of the detritus in the larval habitat. The reason that the Asian tiger is reduced is probably still due to interactions with resident species that hatch earlier, just naturally in the forested area. But more to be done there. Okay. So that's all I'm going to say about the Asian tiger mosquito. This next portion of my talk, I'm going to switch gears a little bit. And I'm going to talk about the work that we do with a couple different species of mosquito in that Gregarine parasite that I mentioned. And this is work that we do at Tyson Research Center. So we're really interested in specifically how climate change and species invasions are going to impact parasite infection. So species invasions can impact infection levels in a lot of different ways. There can be parasite spillback. So like here's an example where a fish has a parasite, okay? And then an invasive species comes in, and this invasive species happens to be a competent host. So now it's infected, and it's spilling out more progeny of the parasite, and the native species actually is now more infected than it was before the invasive species was there. Invasive species can alter native host-parasite interactions by altering the native host density or behavior. So this is work that was done looking at, this is actually done here at Tyson and surrounding areas, looking at how the invasive honeysuckle, you guys all know the terrible honeysuckle that's taking over here. So deer love it, okay? And they bed down in it. It's a good refuge for them. And so areas that have honeysuckle have more deer activity, so it's altering the deer host behavior, which then concentrates more ticks in the honeysuckle, and in fact, they found that it increases human risk of disease. So indirectly, by changing host behavior, we see an increase in disease. And also, the parasites themselves can be the invasive species, or can work in concert with the invasive host. So in the UK, the red squirrel is going extinct because, yeah, our gray squirrel is competing with it for food. Okay, so there's resource competition, but it also came over with a pox virus that's deadly. Doesn't affect the gray squirrel, but it's deadly to the red squirrel. So this is a case of resource competition coupled with an invasive parasite killing off a native species. Okay, so there's lots of different ways. But something that's almost never thought about is the fact that invasive species can actually reduce parasite prevalence, potentially, in native species through what we call a dilution, so it dilutes the parasite. And when we think of invasive species, we think bad, right? That's how it gets the definition of invasive, because it's bad. But there was some literature that showed that if an invasive species comes in and it's not a competent host, it can't make new progeny of the parasite, that it might actually reduce infection in a native species, which would be like a rare benefit, right? So this one paper here, there's a couple of snails in New Zealand, one invasive, one native, and they found that the native had much fewer parasites than the invasive, with the invasive present. Okay, so we were curious, because we've got these invasive species here. Again, to introduce you to our native eastern treehole mosquito that's infected with this native Gregarine parasite, which is species specific, so it pretty much only completes its life cycle in the treehole mosquito. And then we've got this Asian rockpool mosquito that got to Missouri about 2005, so it hasn't been here that long. And I've done some field surveys because I'm a curious kind of person, and I found that there was lots of these rockpool mosquitoes in the woods, lots and lots of them, but I never found them infected. So let me just tell you about the life cycle of the parasite, because this will be important. So this cartoon here, the outer circle is the life cycle of mosquitoes, and inside is the parasite life cycle, which follows. So these mosquitoes ingest the infectious oocysts, they're called, in the larval habitat. They're free-living oocysts in the water. They ingest them while feeding. And then they develop in the gut of the mosquito throughout the life cycle. They fuse as in pupae, and then when the adults close, they shed them out. Okay, so the mosquito has to complete its life cycle in order for this parasite to complete its life cycle. At this 3rd or 4th instar stage, I can dissect a mosquito larva and I can count the number of parasites in it real easily. So I can get determine the infection load, so how many parasites are in an individual, and the prevalence in the population. Okay, so mosquitoes are filter feeding in the water column. This is just some cartoons of them just like shoveling stuff in their mouths and like, just all the amount of water that they circulate through in their day feeding. So if this invasive species, this rockpool mosquito, is going to be common in the water with the native species, the idea is that they're going to encounter these parasites, okay? And if they're not a good host, they might be removing them. So the questions I asked for this experiment are, do invasive mosquitoes reduce infection prevalence in the native mosquito? And what is the mechanism? Are there going to be fewer infections because this invasive species just kind of crowds out and outcompetes the native, so there's fewer of them to keep the transmission cycle going? Or could it be by a reduction in the number of encounters they have with these infectious oocysts? Are they just vacuuming them up and not putting them back out, right? And then thirdly, what is the role of environmental conditions? Because everything happens in an ecological construct, So this was an experiment we did in the field. So the environmental condition we manipulated was leaf detritus. Again, an all-important detritus. So we had high and low amount. And then we had treatments where we removed the invasive species or we didn't, okay? This is what our setup looked like. So this is a much prettier field site than the tire pile I showed you, but sometimes I get to work in beautiful places. So how this worked was a 9-week manipulation. So every 3 days, my students and I went out, we sieved all the larvae out of the buckets, took them back into the lab. Under the microscope, we identified them all and removed all the invasive species in half and didn't in the other. And then later in the day, we put them back in, okay? And then at the end of the 9-week manipulation, we dissected all the 3rd and 4th instar larvae and counted their parasites. And so what we found was what we predicted. Okay, so let me walk you through this figure. This is the proportion of the larvae that were infected. These two are where the invasive species was removed, not removed, low amounts of detritus, high amount of detritus. And we found that when the invasive species was removed, parasite prevalence went through the roof. And when they were allowed to stay, they had, this is very low infection prevalence. So my hypothesis was supported that this invasive species is totally clearing, almost completely, parasite infection in this native species. And that there was an environmental context to it. So when there was low amounts of detritus, they weren't as infected as when they were high amounts of detritus. So there was an environmental context to the impact of this invader. And so the next question was, well, what is the mechanism? This is just showing you that this is the abundance of our invasive species. So just showing you that the removal was pretty good. And then this is the native species. And so there was absolutely no effect of the invasive species on the native species. But look at the difference in the abundance. So there was like 10 times more of the invasive species. So they're taken over. So it wasn't that they changed their density, it was that they removed their infectious particles. Because I did a lab experiment where I put larvae in with oocysts, left them in there for a few weeks, and then counted all the oocysts that were left. And so we are seeing that when the larva is present, there's a lot fewer oocysts. And so finally, and this is the last thing I'll tell you about quickly, is we're also interested in how climate change interacts with invasive species and parasite prevalence. And so we did just like a quick laboratory experiment where individual larvae were just reared in a little bit of water in these vials in the lab, and reared at constant temperatures, 24, 28, 32. This is like 90 degrees Fahrenheit. And we found that temperature is really important for parasite infection. So this is just the number of the parasites that are in the mosquito. And at these 32 degrees, they basically were completely cleared of parasites. So we wanted to take this very controlled condition. So we wanted to take it out into the field and ask, how does the invasive species again, but this time with climate warming instead of different amounts of detritus, how does that impact parasite infection? So with the exact same setup, just instead of different detritus amounts, we warmed half of the buckets 3 degrees Celsius, which is a realistic projection of how much this area is going to warm in the next 30 years. Here's my students. And how that was done, you can kind of see there's an extension cord here. And here's our solar panels. We bought aquarium heaters.
Heating element.
Yeah, they're just little paddles. So they weren't going to burn our larvae, but they consistently raised the buckets by 3 degrees above ambient. And the same setup, removed the invasive 3 times a week. At the end, we took them all in. And we again found, although a less dramatic result this time, this is again proportion infected. These 2, the invasive was removed, heated, not heated. So again, when we remove the invasive, we see that really high prevalence of infection again. And we see they're heated. When the buckets were heated and the invasive was present, we see the lowest amount of an infection. So the take home from here, I'm not going to tell you invasive species and global warming is good, right? But it's difficult to predict because there are things that are unintended, right? And it depends on whose perspective you're looking at from, right?
Benefit or not.
Yeah. So is it good for us that this mosquito that sometimes vectors pathogens is less parasitized? Probably not. Is it good for that mosquito? Yeah. So I don't know, something to think about. Okay. So in conclusion, I won't recap what I went over, but I know that you all are going to ask me what's going to happen in the future with vector-borne disease. It's the $1,000,000 question, right? And I'm an ecologist. I'm not an epidemiologist. I'm not an infectious disease predictor. But my expert opinion, and based on some of the good science that's been coming out and modeling efforts, is that we are going to see range expansion in vectors. And we are going to see more vector-borne disease. At the beginning of my talk, I showed you just even now, as we're still in the early phases of climate change, we're seeing increases. Do I think it's going to be a panacea where we're all going to die? No, I don't at all. Some of the areas that are predicted to see increases in, ticks and mosquito-borne diseases are like Europe, Canada, pretty well-off places. Okay, and we can't forget that socioeconomics and human behavior are really important for vector-borne diseases. So again, if you're in you got this air conditioning, although this is part of the climate change problem, right? And your indoor patio, you're not going to encounter as many infected vectors as if you live in this favela in Brazil where you've got trash laying around. So yes, we're going to see more. They're going to be moving, but the movement we're going to see is going to be in higher socioeconomic areas. And we even know today that, for example, socioeconomics plays a huge role in disease risk. So this was a paper that came out, probably 15 years ago now. There was a big dengue outbreak going on in Nuevo Laredo, which is the sister city of Laredo, literally just separated by this river. And they didn't have fewer mosquitoes in Laredo, but they had almost no dengue, and it was all lifestyle. So that's all I got today. I just want to thank my students who helped me do all this work, and thank you for your attention. Questions? Yeah.
So the length of life of a mosquito is obviously extremely important to whether or not it ever gets a chance to be a vector.
Yes.
So the more they're predated, the less likely they are to get to that point, and
That's a complicated question. And this is why ecology is so important, because ecology teaches us that predation can often produce more adults than without, because you're eliminating resource competition, right? So that's a very complicated question, because it's going to depend on the circumstances. So if you get a predator in there that literally eats everybody, yes, but that's often not the case. So often you'll have a few left over that now grow up huge and strong and are great at transmitting pathogens.
the best of the ones who survive.
Yes.
But I was thinking of birds eating adults.
Oh, I see, I see.
And then you have
Yes.
the decrease in the bird population.
Yes.
And the increase in the mosquito population.
Yeah, that would be a problem. Yes.
And that's not good.
Bats.
They live longer.
Yeah, bats are important too.
Right. They're getting that full
Yes.
lifespan where maybe they wouldn't have 20 years ago
Sure.
because there were more bats, birds, and what have you.
Yeah, that'd be interesting to empirically test, actually. I mean, you wouldn't want to go to a place and remove all the birds and see what happens,
Right.
but yeah, it would be interesting.
It would be hard to control for that.
Yes.
But yeah, if something gets in the water, and then they'd be like hummingbirds. They'd be only the best survive. You've got this super generation of
Yeah.
mosquitoes.
Yeah.
Oh, do any of the invasives, are any of them good pollinators?
That's an understudied area. We know they all pollinate. People have not been very interested in their nectar feeding behaviors because they're interested in their blood feeding behaviors.
They're not worried about the girl.
Yes, yeah. So it's an understudied area of just the amount of how important of pollinators they are.
But they're definitely going from
Definitely.
bud to bud.
Yes. Yeah.
But all mosquitoes are, female mosquitoes are all mammals, sucking, blood sucking, or none of them feed on plants or anything like that. So are mosquitoes.
There is one genus of mosquito that as an adult does not take blood meals because it's a predator as a larva. So it gets all its protein eating other mosquito larvae. But yes, they feed on, but mosquitoes will feed on like lizards and birds and mammals. Yeah.
So if you were gonna say, when you were doing all the things that it was like, you were doing that to those mosquitoes, you were doing that to those mosquitoes, wouldn't it mess up how they evolved?
Yes, it could. But even though, I emphasize how fast mosquitoes evolve, they're not doing it over the span of a summer that I'm doing a project. Is that what you meant?
Like if it was over time, because you sent some over there, and when they come back, or if you research them more, they come back, and I don't know how evolving works, but then they're taking that information and all that, and then over time they evolve.
It could.
Or something they decrease.
Yeah, it could. If you consistently manipulated them for 10 years, 8 to 10 years, you could. Yeah, potentially. Yeah.
So the adaptation experiment that you guys conducted, where you shipped the eggs over, the result is is it the eggs that are adapting, or is it the parent that's laying the eggs that is actually adapting to the parent?
So it's the female that makes the decision to go into diapause, and it's the eggs that actually do the surviving. So I'm not sure.
But in your experiment, the eggs were produced here, and then they were shipped over.
Yeah.
So at the time, the female probably did not make that decision, right?
Yes, it did, because I forced it to, right? So I made the daylight short.
Okay.
So I forced it to decide whether to go into diapause or not.
Just the daylight? Doesn't it need the other conditions, like the temperature and stuff like that?
Yeah, temperature matters. So they were all reared at the same temperature. So that when they were sent out to the field, they had experienced the exact same conditions. And it was just what had happened to the eggs that was going to determine whether they were adapted or not.
Okay. So your experimentation is solely on the daylight, the length of the daylight?
Yes.
Okay.
Yeah. Yeah, and I'm actually getting ready to do another experiment that's very similar to that in design, but instead looking at how artificial light at night in the city impacts their decision to go into diapause, because it's so strongly photoperiodically determined that I'm curious about these half-lit nights that we have, what that's doing to mosquitoes.
That'll be interesting.
I'm excited. I hope it works out.
Are the changes in the egg strictly chemical and biological, or is there something genetic going on too when they make their adaptation?
Yeah, so we know it's genetic. We know it's genetic.
It's
Yeah.
fixed. It's there beyond generations.
Yes. Yeah.
That is pretty fast, isn't it?
Very fast. Yeah. Yes.
And I was thinking the same thing with all of our, wonderful energy-saving LEDs. There's so much more light.
Yeah.
And it's a different
Yes.
light
Yes.
than the old incandescent lights.
Yes.
There's a lot of it.
So these are part of the decisions I'm having to make right now. What kind of lighting do I give them? The old, lights that used to be in our street lights, or the new LED lights? Like, what do I choose? So these are all really good questions.
More light at a much different
Yeah.
frequency.
Yeah.
Because one of the things with the high and low pressure sodium was that orangeness
Yeah.
wasn't delivered. I mean, we saw it as visible light, but what we have now, it's a lot more light.
Yeah. It's all really new territory to explore.
And I'm sure it's affecting them.
I believe it is. Fingers crossed.
I think you're okay. It's a lot more light.
Yeah.
And we tend to leave it on longer because it uses less energy. We're not as sensitive to shutting it off.
Yeah.
Shut that billboard off at midnight now.
Yeah. Anybody else? No? All right. Thanks again.